I'm Allison.
I'm Kelsey.
I'm Natalie.
I'm Lauren.
And I'm Kendall.
And we're Group 1, and our presentation today is over the CRISPR technologies. Has science really gone too far? As knowledge and research continues to excel and discover new and efficient ways to cure diseases and illnesses, ideas and expectations tend to expand as well. One of the newest discoveries and phenomena going on in our present-day society is called CRISPR. CRISPR allows scientists to edit genes very specifically. It is a tool that is able to find breaks in the DNA, cut it out, and then repair the DNA genome. For those of you that are not aware, a genome is a complete set of an organism's DNA. Each genome contains all of the information needed to build and maintain the growth of an organism. It is almost like your body's own instruction manual that allows your body to grow and function each day. In humans specifically, there is a copy of the entire genome in every single cell that has a nucleus. That is more than 3 million DNA-based pairs, and if you're trying to wrap your head around that, it could fill 200 500-page telephone directories. CRISPR is able to find each of these genomes and manipulate the DNA sequences within them. This tool is made up of a protein called Cas9 that is accompanied by a guide RNA. This is what keeps the Cas9 on a leash and is able to go find those specific breaks in the DNA. In the health industry, this is a huge advancement because it could potentially take away most genetically linked diseases, breaks in DNA, and errors within the genome. To put all of this into perspective, take for example, if CRISPR was on a genome of a mosquito that carried malaria, it could cut out that disease. This could lead to a chain reaction within the environment. All of these mosquitoes that have been genetically modified are then released into the world, and they are free to reproduce with any of the mosquitoes. This could result in all of the mosquitoes turning malaria free. With that being said, this could affect the amount of people with malaria as well. If CRISPR is eventually published and proven, will this have a positive or negative effect, or both? Will people start volunteering their bodies for research? Another topic that could arise is designer babies. For those of you that don't know, designer babies are modified while they're in the embryo to have specific genetics and genes that are determined by their parents. Should there be requirements to qualify for CRISPR, or should everyone have equal rights when given the opportunity to use CRISPR? Should or could society run into issues if everyone starts to design their own babies because of CRISPR? Or would the society turn up for the better because CRISPR is able to repair the genes and have potential results in curing diseases and genetic illnesses? As a group, we view this advancement as a positive contributor to the health industry, as well as the lives for Americans. We feel as if CRISPR is a huge oncoming advancement, but should be highly regulated. The human population is not ready for CRISPR to start regulating the human genome, and more information and research is needed. They have not yet done any research over the CRISPR in a human yet. CRISPR still has plenty of off-targeted cutouts, and it is not specific enough to be used in the complex human genome. Although CRISPR could be the start of curing genetic diseases and DNA breaks, it is not ready to be used in the lives of human embryos because of the potential dangers and implications that we are still unaware of. We also think CRISPR needs to be very tightly regulated to prevent usage abuse throughout the environment and population. The CRISPR-Cas9 system has a great promise in broad applications such as stem cell engineering, gene therapy, tissue and animal disease models, and engineering disease-resistant transition plants.
Now we're going to talk to you a little bit about how CRISPR actually works. Here you have a guide sequence and a guide RNA. A genetic sequence called the guide RNA matches with the DNA they want to modify.
Then, Cas9 comes in, and the sequence is added to the cell with the protein, which acts like scissors to cut the DNA.
Once the DNA is cut out, the RNA and Cas9 leave the scene.
Another piece of DNA is swapped in the place of the old DNA. Enzymes repair the cuts, and
bam! You have yourself some genetically modified DNA.
Natural gene mutation is how we as humans grow and develop. Mutations can be either categorized as germline or somatic, meaning they were either acquired as an embryo or after. Very few mutations that occur within our body are bad for us. In fact, there are many mutations that take place that either go unnoticed or are beneficial for us as humans. The development of these mutations have helped form Darwin's theory, survival of the fittest, as well as natural selection. The fittest surviving is what has accelerated our growth and development of human life. CRISPR is not necessary for this human development. Natural selection takes place for a reason. CRISPR allows scientists to intervene and disturb this process. The interruption of natural selection may lead to a future of designer babies or a genetically modified new human race. As Allison previously mentioned, human genetics is incredibly complex. In fact, there is an estimated 3 billion genes. That's huge. This number makes it impossible for scientists to understand them all. In fact, new data is published every 24 hours on new findings about genes and genetics. This fact alone shows how unlikely it is for scientists to really grasp the idea behind the genome. It's so easy for scientific error to take place. All of this information leads up to the big question of what catastrophic effects CRISPR could have if we utilize its abilities for human embryo gene modification. The argument for gene modification is that it will protect our future generations against genetically inheritable diseases. The good news is this technology to accomplish this is already available without using CRISPR and gene modification. Today, IVF treatments allow for the fertilization of various eggs at once. Once the eggs have been fertilized, scientists can run a PGD test to scan for various genetic diseases. This process gives us the means to reduce the occurrence of genetic disorders without playing the hand of God. If we have the means to do this now, then why should we continue to develop CRISPR for human gene modification, if it may lead to the potential prevalence of designer babies? Some members of the scientific community believe that moratorium should be called on CRISPR due to the threat of designer babies and the potential of a eugenic society. Others, however, believe that by terminating CRISPR's research, it would be very unethical because you would also be terminating research that could lead to the development of new vaccinations or medications against various bacterial diseases. The scientific community needs to meet in the middle with these two viewpoints. CRISPR does not need to be stopped, but it does need to be strictly monitored and limited. If embryo gene modification develops, an infinite amount of ethical controversies will arise. One of the most important questions to ask is if gene modification is put into action, then who will have the authority and jurisdiction of this process? Will there be a global onset of rules and regulations, or will each country have its own unmonitored jurisdiction of CRISPR and its possibility to create designer babies? If each country does have its own authority, then we need to fear the potential of designer babies. Will power-hungry rulers or dictators use CRISPR to develop their own genetically enhanced population? If so, this new population could be genetically modified to be stronger, more precise, and larger. Or will they completely alter the various organisms, particularly humans, to complete genetic makeup maliciously? Who knows where this would leave our world? Or would other countries, such as our own, let vanity interfere with science through the use of CRISPR? This would happen without a doubt. The vain seeking out a perfect baby would come at a cost. The socioeconomic status that we have in society today would completely shift. An even larger social hierarchy would form, and the human worth would devaluate. How would you feel knowing what makes you a unique individual would soon be looked down upon? Our personal traits and characteristics may soon form to be obsolete. What if your future children looked like strangers, people that you didn't even recognize? Or imagine not having your dad's eyes or your mom's hair. Or even crazier, what if your next door neighbor's child looked like their favorite celebrity because their parents wanted to use CRISPR to modify their child's looks? These are all very real consequences that would happen from CRISPR and the effects of designer babies. Finding the solution of limiting CRISPR's use will avoid all of these future problems we all fear. The limitation of this technique will allow for us to retain family traits that have been passed down for generations. It will also allow us to feel unique without losing our individual self-worth. While these negative aspects will be avoided, CRISPR will still stand to be utilized for scientific advances that are beneficial for our populations and generations to come.
Although the CRISPR is revolutionary technology, it isn't ready for human models just yet. It needs to be researched and understood more and then regulated to avoid abuse. We just don't understand enough about epigenetics, specifically how editing human embryos and specifically the germline mutations, which are the heritable traits, would affect future generations. It's a very slippery slope towards unethical or unsafe uses of this technique. The solution would be to have a specific legislation to regulate the use of CRISPR to limit and prohibit the use of germline cells to avoid designer babies and keep the system used with diseases and other scientific advancement to better all of humanity. One way that CRISPR has been abused already is through a Chinese study, a study that involved non-viable human embryos that resulted in non-edits that were off-target with pretty low efficiency. Off-target is when the CRISPR-Cas9 system acts on genes it is not programmed to act on. So the research team found a surprising number of these off-target mutations, which assumed to be introduced by the CRISPR-Cas9 complex acting on another part of the genome. This effect is one of the many, many safety concerns surrounding germline and gene editing because these unintended mutations could be so harmful. The rates of such mutations were much higher than those observed in gene editing studies of mouse embryos or human adult cells. The head researcher even noted that his team likely only detected a subset of the unintended mutations because their study looked only at a portion of the genome known as the exome. If they did the whole genome sequence, they would likely get many more. There is also the example of gene drives and pushing the misuse of the CRISPR system. Gene drives can force a trait through a population defying the rules of inheritance. This leads to the genetic changes throughout an entire species instead of being the 50-50 chance of a trait being passed along to the next generation. A gene could now be pushed to 100%. The ethical implications of this are unknown changes that it would make to our ecosystem. We are attempting to make changes to species that would ultimately change parts of the natural world that we have no control over, and we have no idea what implications this would have. The researchers have used gene drives and used the CRISPR system to drive a gene through a species. The researchers who have used gene drives use the CRISPR system to drive a gene through a species. The CRISPR system is flexible and works in almost any species so far, and this gives the researchers the ability to drive any gene they want to and that they know how to alter. Gene drives are very useful for genetic engineering and are really pushing science forward, but they are so dangerous because they are cross-generational, which means that they could affect other generations of other species and so forth. This technology could also suppress genes that are essential for life and effectively wipe out a whole population or species, which is so terrifying. These real-life examples are why we should be so wary of these gene editing programs because of how easily they can be manipulated and abused, and why it is essential that we put legislation to regulate them into place. The NIH, the National Institute of Health, has already refused to fund gene editing experiments on embryos, but there is still tons of private money pouring into CRISPR, and there is the allure of fame surrounding whoever creates the first designer baby. We are not saying that CRISPR should be banned because it is absolutely revolutionary technology, and it is truly pushing the speed of science and offering advancements that are under-talked about because they are so overshadowed by the fear of abusing this technology and creating a world full of designer babies or potentially damaging our ecosystem by changing the traits of a species completely. CRISPR has also been studied as a replacement for antibiotics, drug developments, and has a lot of potential as a tool for treating a range of medical conditions that all have a genetic component, including cancer, hepatitis B, or even high cholesterol. These are all groundbreaking reasons to keep using CRISPR, as it is catalyzing the advancement of science. But if we are to keep using it, we must implement regulations so that we can continue to advance without destroying ourselves by unethical practices that are foreign and potentially dangerous.
We do know quite a bit about CRISPR, and hopefully you're getting the gist of it. But one of the scariest and most dangerous things about this technology is how much we do not know and what it has the power to do. I'm going to quickly revisit and illustrate what CRISPR does and how it works in order to show you what CRISPR has the potential to do in the future. The CRISPR-Cas9 system consists of two key molecules. one of which is an enzyme, Cas9, which acts as what one would consider a pair of molecular scissors that cut two strands of DNA at a specific location, thus breaking the DNA helix structure. These scissors are guided by a piece of RNA, which tells Cas9 where to make these cuts. These cuts are made and the DNA is damaged, and when that occurs, the cells essentially go into a panic mode and must be repaired immediately or as fast as possible. CRISPR-Cas9 technology can repair or put the structure back together by either taking an entire gene out, by putting one in, or by editing a single letter in the genome. It may help to think about this editing system much like you edit a Word document. You skim the Word document, and once you find a portion, word, or even letter you would like to change, you plop your cursor down, backspace, and make the change. You have the ability or power to add sentences, remove mistakes, add or delete letters, all in order to tailor the document to be exactly how you desire it. Editing a Word document is safe and ethical, obviously, but it is editing a human. With CRISPR, we have the power to make any edit we wish. But should we? Are we truly ready as a society to handle all it can do? Natalie spoke to you moments ago about the concept of gene drives and the ability to push a trait through a population. Mendelian genetics essentially states that there is a 50-50 chance of a trait being passed along. However, a CRISPR trait is 100% heritable. But how? CRISPR inserts not only the new gene, but the cutting and pasting machinery that allows it to make its edits. Therefore, it guarantees that the CRISPR gene will get passed on. In humans, when used on germline cells, both chromosomes will become equipped with the machinery, making heterozygous traits homozygous. Now picture this. Imagine someone creates a gene drive and pushes a trait that suppresses an essential trait for life, such as the ability for males to reproduce. It would take centuries to push the gene, but eventually, and inevitably, all males would possess this gene. This would obstruct the ability to procreate and ultimately lead to the eradication of our species. This idea may seem far-fetched or unrealistic, but this is the harsh and terrifying reality of what CRISPR technology is capable of. If this technology isn't highly regulated, it could easily be abused and has the potential to be detrimental to not only our species, but our entire ecosystem. If that doesn't scare you, then maybe this will. Jennifer Doudna, co-founder of CRISPR-Cas9, has called for global pause in any clinical application of CRISPR technology in human embryos. That's right. The inventor herself is requesting that we step on the brakes and really think about the ethical and societal implications of the technology, as well as the unintended consequences of its uses and the intended impacts. Doudna, who knows more about this topic, addresses there are still a great deal of unknowns. We need to learn how to control and limit the effects of target DNA cutting, as well as the dangers in cutting the DNA to begin with. The DNA must first be damaged to be edited and repaired, but what if this process goes wrong? Doudna admits there's so much more to learn, and that this technology just truly isn't ready to use on viable human embryos. She also understands that the CRISPR technology, the idea of living in a genetically modified world with desiring babies, is no longer science fiction. The technology was intended for therapy use, but will we take it too far and use it for physical enhancement? That is why we believe that we do in fact need to put a halt on the use of CRISPR in human embryos and make sure there are strict regulations and limitations of the uses of this technology. We also need to think deeply about the ethical and societal impacts that it will have.
The scary truths about CRISPR may overshadow the good that it is bringing to drastically improve scientific research today. While CRISPR has the ability to edit literally any gene that one would want to suppress or strengthen in any organism's DNA, it can also change the outcome of its overall appearance and its potential offspring. Today, our society is infatuated with not only oneself's appearance, but others as well. The Kardashians lead the world in changing their appearances almost daily. From lip pumping, teeth whitening, wig wearing, butt implants, and many other procedures that one improves one's body, they almost encourage you to change who you are and love what you will become. Have any of you seen the before and after pictures of Kylie Jenner? If you have not, you won't recognize the girl she used to look like. Kylie is growing wildly successful with her new makeup line and is also a role model for young adults and children. Years from now, if designing babies become a reality and an expensive option when you go to see an OBGYN,
those girls who look up to Kylie can pick and choose traits to have babies that look identical to who Kylie is now. Natalie talked about possible solutions to stopping designer babies by way of using CRISPR is to heavily regulate who can use CRISPR and utilize its easy and precise mechanisms will be key in halting the future of designer babies and testing on humans. China showed the world what happens when CRISPR is abused and just how much we don't know about CRISPR. Because of their experiment, it showed how easily CRISPR could be used in negative and destructive ways without even trying. Remember when Lauren said even the co-inventor of CRISPR, Dr. Jennifer Doudna, believes the funding of CRISPR should be paused because of its easy-to-use system and cheapness that could allow almost anyone to use and abuse CRISPR? The power of CRISPR is not completely understood by scientists yet, but continues to be funded and researched heavily as the race to fame drives ambitions past all ethical solutions. Giving the power to the government and making strict laws and regulations may be one solution, but who will be responsible for making sure these regulations are governed at all times? The conceivable effects of CRISPR draw large and horrifying consequences if no solution is put in place in our society preventing CRISPR to be used as on humans at this time. Health Expectations article, What Choices Should We Be Able to Make About Designer Babies, says that CRISPR as a way of improving public health is highly accepted among the public, and most of those people believe that CRISPR's ability to cure cancer and create new vaccines for incurable diseases supersedes its ability to edit one's embryonic appearance. In 2002, the Genetics and Public Policy Center at Johns Hopkins University completed a survey on reproductive cloning, designer babies, and genetic modification, and resulted in over 70% of respondents saying that they were against using said genetic modification techniques to interfere with more complex human traits, such as intelligence or physical strength, but were in overall favor for gene modification improving public health. We believe that CRISPR is an extremely useful method in organism genome modifications pertaining to vaccine development, fighting antibiotic resistance, ending genetically linked diseases, fixing errors with the genome, and much more. But the dangerous capabilities of not only modeling one's baby to desired specifications and many other treacherous possible outcomes was a very concerning matter in our materialistic, fast-paced society. The power of CRISPR could put an end to a whole species without proper research, understanding, or professional scientists. CRISPR has potential that is far beyond designer babies, but who is to say that the power to design your own baby will not become the hottest trend in years to come and the most sought-after prize today? Could you imagine a world with no limitations to editing one's genes, or you could design your own baby to look like Blake Lively or Ryan Reynolds? Now that Allison, Kelsey, Natalie, Lauren, and I have told you about CRISPR and the potential detrimental effects of designer babies, it's your turn to consider what your future will look like. While the future is inevitable, it is our decisions now that make our future what it will be. So what will your decisions be? The future of CRISPR will make an imminent difference in public health and scientific research, but it's up to you whether or not it will change the world for the better or for the worse. Say no to designer babies and testing on humans. Yes to CRISPR's research. Thank you.